Hello all, welcome. In this video we are going to see about CSIR Net previous year question concept series of C Elegans. And you are watching this on Life Science Library. First question from November 2020 CSIR Net question on SK1 protein concept. Now we are going to see what is this SKN1 protein concept. So consider zygote of C. elegans. It forms two different categories. It can be AB, it can be P. P is a stem cell, P1. AB is the founder cell. It is conditional in specification. It is autonomous in specification. Okay. ABA can be divided into ABA and ABP. P1 can be divided into EMS and P2. So this SKN1 protein, what is the function of this SK1 protein? This SKN1 protein will induce this P1. This is the basic function of SK1 protein. Now there is other signaling system as well. P1 is divided into EMS and P2. P2 is divided into two things. That is, EMS is divided into MS and E is divided into C and P3. Okay. Now, MED 1, MED 2. All these things will help SKN1 and to stimulate PK1. And these things will inhibit PAL1. Function of this PAL1 is to stimulate P2. PAL1 is being inhibited by MEX3. This is the SKN1 signaling system. Other important thing to note about is what are these EMS. So E codes for this intestine, M codes for pharynx, hypodermis, C codes for again intestine. P3 will be divided into two different categories. That is P4 and D. Okay. This C codes for muscle, D codes for muscle, P4 codes for germ layers. So this is how the progression occurs. Now we are going to see about SKN1 mutation case. This is the concept. So in C. elegans, SKN1 protein controls the fate of EMS blastomere which generates posterior pharynx. With reference to the above, which of the following statement is incorrect? MS blastomere is able to generate pharyngeal tissue in isolation. Correct. Because they are autonomous. Embryos from SKN1 deficient mothers lack both pharyngeal mesoderm and endoderm derivatives. Correct. SKN1 activates MED transcription factors whose level of activity controls the fate of EMS lineage. Correct. So SKN1 is somehow associated with MED. They help in controlling EMS. Where in EMS it is divided into, I said it is divided into MS and E. This MS is autonomously specified, but E is conditionally specified. Okay. Then, next point. Embryos which are SKN1 null mutant. Null mutant in the sense there will be no effect of that mutation. Will make extra embryo hypodermal. How it will make extra hypodermal skin? Because the mutation will not be expressed since it is null mutant. Null mutant. Okay. So C is the correct answer. Same question. Now, hedgehog pathway of November 2020. We have already uploaded a video on sonic hedgehog signaling system. Please go back and watch this video before answering this question. That will be of great help. So this question does not need concept explanation because concept is already explained in our channel itself. Hedgehog pathway is extremely important in vertebrate limb development, neural differentiation and facial morphogenesis. In accordance with the above statement, what could happen in mice that are homozygous for mutant allele of sonic hedgehog? So homozygous mutation. Homozygous mutation of sonic hedgehog. They will form eye in the midline. This condition is called as cyclopia. Midline of the face could be reduced and a single eye could form in the center of forehead. So B is the right answer. Next question from November 2020, again from sonic hedgehog signaling system. Sonic hedgehog specifies anterior posterior axis during limb development. Which one of the following statement regarding is correct? Hedgehog secreting cells undergo apoptosis after performing function. No. Descendants of hedgehog secreting cells become bone. No. When genes of hedgehog and GLE3 are conditionally knocked out in the mouse limb, resulting the limbs do not form any digit. No. Specification of digits is primarily depend upon the amount of the time hedgehog is expressed to small extent in the concentration of hedgehog protein that still receive. So for 12th question, D is the appropriate answer. Next question from December 2018. Zygote of C. elegans exhibits rotational cleavage. Learning. Rotational cleavage. And the rotational cleavage we have seen in previous video. That is humans. Mammals have rotational cleavage. Okay. When the two blastomeres formed, that is P1 and AB, are experimentally separated, which of the following outcomes may be possible? Okay, we know AB undergo conditional specification, but P undergo autonomous specification. Let's see what is the answer. P undergo autonomous specification and AB undergo conditional specification. Here is the right answer. We have seen this explanation in the first question itself. Now, 46th question regarding paracrine signaling. Following are the assumption, consider this diagram. This diagram is present in the question. Following are the assumptions derived from the given statement. Medium A contain FGF and PDGF. Medium B contain retinoic acid. Cells are cultured in the medium B and were determined to become functional neurons prior to the addition of the medium. No, they are stem cells, so they are not conditioned before the addition of morphogen. So C is not a correct statement. Remove all the statement that has C. A is the right answer. June 2018 question from C. Elegans. So this is about C. elegans specification. C. elegans will have an anchor cell. Is called as anchor cell. Wait, my handwriting. Now we are going to see determination of valval development in C. elegans by the help of various signaling system. Okay. First consider this cell as an anchor cell. Okay. Now consider P3P, P4P, P5P, P6P, P7P, P8P. Okay, wait for a minute. P3P, P4P, P5P, P6P, P7P, P8P. Okay, this anchor cell present in the C. elegans will produce the signal. It will directly reach to the central cell. It is length 3. Length 3 will reach this P6P in high quantity. More signal will reach the center cell. Okay, we have darkened the signal. More signal will reach the central cell. Okay, so it conditionally specified to become primary cell. Only less amount will reach P5 and very less amount will reach P7. So they will specify to become secondary cell, that is lateral cell. Length 39 help in specify valval cells. This is an important pointer. Length 23 act as a receptor present in the all valval precursor cells. They will accept this length 3. Another important thing is, so we have P6P as primary, P5P as secondary, P7P as secondary. This is primary, this is secondary, this is also secondary. P6P will release length 12 to both the neighboring cells to prevent them from becoming primary cell. This kind of signaling is called as lateral inhibition. Okay, this kind of signaling is called as lateral inhibition, where the signals, that is length 12 produced from P6, will inhibit P5P and P7P for becoming primary cell. Okay, this is the concept. Now let's look into answering this question. Following statements are made regarding valval development in C. elegans. Valval precursor cells are influenced by the anchor cell to form equivalence group. We have drawn all the things from P3, P2, P8, P. All these things are equivalence group in the sense all the cells have the potential to become primary cells. A is correct. Loss of function. Mutation, Lentwell mutation, both the cell become uterine, whereas the gain of function mutation, both the cell become anchor cell. Other two condition is anchor cell produces lag and Len 12. It form uterine cells. The lag 12 will give rise to lag 2. This lag 2 act as a protein. So these are produced by chance event. BCE from C. elegans as two different categories by delta notch pathway. This act as a protein. This act as a receptor. So, consider this as a concept. When Len12 is not present, when the Len12 is mutant, uterine will be formed? No. When the Len12 is mutant, both the cells become uterine, whereas gain of function mutation both become anchor cell. B is a wrong statement. If anchor cell is destroyed in the early development, all 6 valvular precursor cells divide once and they contribute towards the formation of hypodermal cell. Correct. Anchor cell, ventral cell, uterine precursor decision is due to delta knots mediated by the mechanism restricting adjacent cell. Yes, we have seen now. Paracrine factor secreted by the anchor cell directly activates notch delta pathway. No. Why? Because consider wild type C. elegans. In wild type C. elegans, Z1PP form lag 2 that act as a notch. Z1AAA C2 that formed Len12. Okay, give rise to uterine, and they have notch. Notch is a receptor. बात करें so A is the right answer. Coming to December 2017 question. We have seen all these things. Overexpression of Len3. What will happen if Len3 is overexpressed? P5P, P6P, P7P all adopt primary fate. Okay. Now, reduced function of Len3. Len3 is reduced. P6 adopt primary fate and the rest adopt tertiary fate. Okay. Loss of function of Len3 and gain of function of Len23. They give rise to multivalva. So I have said what is Len3, what is Len23. I have made a mistake. No, correct. Let 23. So what are the signals here? Len. So what are the signals here? Len 3. It's not Len 23, it is Let 23. It is LET 23 that help in the specification of valval precursor cells. Okay. बात करें. Please do correct this. Let, it is LET23. Okay. So loss of function of LEN3 and gain of function of LET23 leads to multivalva. Loss of function of LEN3. If LEN3 is loss of function of LEN3, all PVC adopt tertiary fate. Okay. This is the correct answer. Now from December 2017. C. elegans embryo uses both autonomous and conditional mode of specification. Conditional specification at 4 cell stage can be seen in the development of endodermal cell lineage and also in the establishment of dorsal ventral axis. Following are the few statements regarding this. Okay, let's read this statement. If P2 of the cell is removed at the early 4th stage, EMS will divide into 2 MS cells because MS cells are what? MS cells. Just now we have seen in the previous questions. What is MS cells? बात करें, शुरू करें. MS cells. Wait. Okay. So EMS cells are conditionally specified to give rise to 2 cells, that is MS and E. E is conditionally specified. CMS is conditionally specified, but MS is autonomously specified. MSA, that's what you can remember. Okay. So we know P1. Stem cell. EMS. Then P2. We have a signaling system. We know about SK1. We are not going to see about SKN1 and PAL1. We have seen in previous question. Now we are going to see about delta notch pathway here. These two have a signaling system. It has MOM 5 and P2 has MOM 2. They involve in the delta notch signaling system. Okay. So if they are asking, if P2 is removed, what will happen? MS will form because they are autonomously specified. They don't need this kind of signal transduction. E is conditionally specified. So E will not be formed. That's it. If P2 cell is removed at the 4th cell stage, EMS will divide into 2 MS and no endoderm will be made. So what is this part of this statement? What is this? Why they are saying that no endoderm is made? MS will give rise to, if EMS cell is removed, MS will give, there will be 2 MS, no. Each will give rise to mesoderm. Okay. Otherwise, if P2 is not removed, consider a case, wild type case. If P2 is not removed, EMS cell will give rise to MS and E. MS will form mesoderm and E will give rise to intestine, that is endoderm. If at all, if P2 wild type is not there, P2 is removed, what will happen then? So this MS is autonomously specified. E is conditionally specified. So E will not be there. There will be, instead of E, there will be another MS. So endoderm will not be produced. They give only rise to mesoderm. Understand? Understandable? Are the statements understandable? Okay. I hope it's clear. First statement is correct. Next, coming to another concept of POP protein. EMS will give rise to two things, that is POP1, POP. In a case of MS and E. In E, POP is deficient. In MS, POP is high. Okay, are the things clear? POP1 deficient embryos, both EMS daughter cell will become E cell. Okay. When the position of ABA and P are reversed, the fate gets reversed and no normal embryo formed. No. C is the wrong answer. So we will understand this concept of ABA and ABP. This concept of ABA and ABP. So consider this. ABA, ABP, P2, EMS. EMS, P2, ABP and ABA. Okay. बात करें. So the P2 will give rise to APX. Will have GLP1. Again the signaling is delta notch. Here EP2, EMS, ABP, ABA. This APX will go and attach to ABP. This is a kind of signaling system understanding between stem cells and founder cells. Okay. It is in delta notch pathway. Now coming back. Position of both ABA and ABP or reversed. The fate get reversed. No. Embryos whose mother have GLP1 mutant. ABP is transformed into ABA. Okay. So, these right answer. A is the correct statement. Okay. All three are wrong. Okay. Making it clear. A is the correct statement. शुरू करें. GAY 2017 paracrine signaling system. So we know what is hedgehog, hawks. Hawks we have not seen. We will be seeing in the next video. Hedgehog signaling. Hedgehog signaling is related with what all the things. Now we have seen about cyclopia condition. So P is 3. WNT signaling is associated with Fessel receptor. Okay. Q R2, P3 R2. So just guess what is the answer. This is the answer. Now, not signaling. Involved in the signaling at 4-cell embryo stage in C. elegans through GPL1 expression. Now, not just now we have seen that. Then, Hox protein. Hox protein is required for disease formation. They are required for lymph development. TBX is one of such things. They are required for that expression. This is the correct match. December 2015. Question. C. elegans and anchor cell, a few hypodermal cell taken in part of vulva. Experiment performed to understand the role of these cells in vulval formation, and the results obtained are as follows. Anchor cell is killed by laser beam. Hypodermal cells do not participate in the vulval formation and no vulva develops. 6 hypodermal cells closely associated with anchor cell is called vulval precursor cell are killed. Valval precursor cell. If anchor cell are killed, no vulva develops. Okay. If 3 central cell vulval precursors are destroyed, 3 outer cells which normally form the hypodermis take the fate of vulvar cell instead. Okay. Now study the statements given. Anchor cell act as an inducer. Correct. Well, correct statement. 6 hypodermal cell with potentiality to form vulva form an equivalence group. Okay. 3 out of 6 hypodermal cell participate in vulval formation. Central cell function as primary cell. Is that a center? Is that present in the center? We call it a central. Is that present in the center? Go and check out. Then, primary cell secretes short-range juxtacrine. Is that juxtacrine? So, what are the types of signaling? Juxtacrine, then paracrine, then endocrine. Okay. Juxtacrine, paracrine, endocrine. We will see all these when we see about signaling systems. Okay. A, B, C are the correct statement. Check out whether just now we have seen about P6P, P5P and all those things. Check whether it is center. Whether it is in center. A, B, C are correct statement. A is the right answer for December 2015 question. Question number 133. A is the right answer. June 2015. C. Elegans. Bilkul. Fatafat, try answering this question. An anchor cell was violated in early development, no vulva formed. Correct. Dominant negative mutation of let23. Primary vulva formed, but secondary vulval formation does not. Let's keep this. Cell adopting primary fate inhibits the adjacent cell by Len12, not Len39. C is wrong statement. We'll remove C from the game. Constitutive signal from hypodermis inhibit the development of both primary and secondary fate, but it is overruled by initial signal from the Bilkul correct statement. So, now coming to B. Dominant negative mutant of let23. Let23, what is the present of meaning of let23? They act as a receptor for all the signal from anchor. If that is absent, that is mutant, then the other things will not occur. B is a wrong statement. So, clarifying, C is the right answer. December? Okay. Yes. December 2012. C. elegans during embryogenesis anchor cell and 6 hypodermal vulval precursor cell get involved in the formation of vulva. 3 of the hypodermal vulval precursor cells are killed by laser beam. A normal vulva is still formed. It is due to the possible reasons. What are the possible reasons? Read the second statement. When 3 hypodermal vulval precursor cells are killed, 3 neighboring hypodermal non-vulval precursor cells are freshly recruited. No. Galat statement. Wrong statement. We can remove these two from our game. Anchor cell acts as an inducer which can spatially induce only 3 hypodermal cells from vulva. Only? Is it a correct one? Okay. If only is present in any of the given statement, please do mark it and think it over before answering. This is a kind statement from my side. Okay. Anchor cell functions as an inducer which can induce epithelial cells of gonad to get recruited to compensate for the loss. Or 6, now read the first statement. 6 hypodermal vulval precursor cells form equivalence group, out of which only 3 participate in the vulva formation and 3 remains as a reserve cell. When comparing these two, C and A, which can be the possible answer? Epithelial cell getting recruited in the gonad to form vulvar precursor cell is a big game. Okay, don't get complicated. D is the right answer. D is the correct answer. See, another thing from Drosophila melanogaster. Let's recall and let's recall again and again. Drosophila melanogaster. 3 embryos. X, wild type. Okay. Y, mutant for bicoid. Okay. Z, mutant for nanos. Okay. All these 3 things were injected with bicoid in their posterior pole. Injected with bicoid in the posterior, injected with bicoid in the posterior. Okay. What could be the phenotypes in the resulting embryo? If bicoid is injected here in Z, what it will form? There is no nanos. In the place of nanos, bicoid is injected. So it forms 2 head, no tail. Okay. In bicoid mutant, bicoid is injected in the posterior. 1 head will form. In wild type, already a bicoid is present. So it forms 2 head. It forms only 1 head. That too in the posterior side. Okay. Now we go and check the statements. Embryos of X and Z, these 2 cases, will form head on both anterior and posterior side. Okay. While embryo Y will develop head only in the posterior side. B is the correct statement.